Okay, I'm going to start my presentation now. Hello, my name is Aja. I am serving as the Director of Biology and Ambassador of Illinois IU STEAM Advisors, which is bringing you this presentation series on a range of topics in biosciences. Today, I'm going to overview genomics. I'll start by describing what genomics is and how it is different from genetics, and then I will also explain the difference between functional and structural genomics, and then I'll explain how a genome defines who we are and what codons, introns, and exons are. I'll also briefly overview what gene mapping is and how it's different from gene sequencing. I'll also then talk about the relation between genomics and diseases, and also talk about gene editing. And then lastly, I will end by describing how AI will help in the future of genomics. So I'll start with the first topic, which will describe what genomics is and how it is different from genetics. It is important to understand the rule of the words to fully comprehend their meaning. So the word genomics originated in 1986, coined by geneticist Thomas H. Roderick. So he proposed the name for a new scientific field focused on the study and comparison of genomes across species. The term combines genome with YSA suffix, which refers to the application of skills and techniques. The genome would come from genomics, which was coined by Hans Wilkner in 1920, which is merging the Greek word genesis, which means origin and birth, and soma, which means body. So this means the body of genes. Our genome is the operating manual containing all the instructions that help us to develop from a single cell into a human being. It includes the complete set of genetic information. This information is held in the deoxyribonucleic acid, or DNA, which is packaged into structures called chromosomes. Each chromosome includes stretches of genetic code, which are called genes. Humans have about 25,000 genes, and each one gives an instruction to the body. Humans usually have 2 copies of each gene. 1 copy is inherited from each of the biological parents. There are usually 23 pairs of chromosomes in each cell in the body. The entire amount of DNA in the cell, including all 25,000 genes and the known crime part of the DNA, it totals to around 6 billion genes, plus the non-genetic information, and all of this is called the genome. Another analogy to understand it would be that if the genome is an instruction manual, then a chromosome would be a chapter, and genes would be sentences in the chapter, and DNA represents the words that make, the letters that make up each word. So now let's understand the difference between genetics and genomics. Genetics is the study of genes and their effects one at a time. Genomics is genetics on a larger scale, the study of many or all of the genes. DNA is the molecule of life containing the information that defines and makes us our cells. In eukaryotic cells, like human cells, most of the DNA molecules are stored in the nucleus, organized into chromosomes. Some DNA material is also stored in the mitochondria of the cell. I will discuss the structure of the DNA molecule and how it is organized in depth in the next topic. So now just remember that genomics is the entire set of DNA information. This information consists of both the coding and the non-coding part of DNA. The coding part of DNA is what is termed genes, and its study is genetics. Therefore, genetics is the study of heredity in an individual. Structural genomics focuses on the physical structure and organization of genomes, including sequencing and mapping, while functional genomics investigates the gene expression and function within a genome. Essentially, structural genomics provides the blueprint, while functional genomics explores how the blueprint is used. One way to understand is that structural genomics is how books are organized on a bookshelf, and how functional genomics is how a book can impact a person, and like their how a book can impact a person. So now we'll understand why the study of genomics is so important. So to understand this importance, you first need to understand how the genome is structured and what coding and non-coding parts of the DNA are. We'll first see the structure of the DNA molecule. Remember, the DNA is a double helix structure that contains the genome and is stored in a highly coiled form in the 23 chromosome pairs in humans. A DNA molecule is composed of two long strands, each of which is made up of building blocks called nucleotides bonded together. Every nucleotide is made of three components: a phosphate group, a deoxyribose sugar, and a nitrogen-containing base. Nucleotides are all identical except for their base. DNA has four different bases known as adenine, thymine, cytosine, and guanine. Bases are often referred to with the letters A, T, C, and G. The two strands in DNA are held together by hydrogen bonding between the bases. DNA bases always pair the same way: A with T and C with G. The nitrogen bases are like Lego pieces, where A can only combine with T and G can only combine with C. The double-stranded molecule is twisted into a double helix shape resembling a twisted ladder, where the rails of the ladder are made up of the phosphate and deoxyribose sugar, and the rungs of the ladder are made up of nitrogen base pairs. Opposite strands run anti-parallel to each other, meaning that they run in opposite directions. This ensures that the strands fit tightly together. Our genome can be divided into two parts: the parts that code for proteins and thereby for the traits and that are called genes, and the part that does not code for proteins, also called non-coding DNA. The intergenic regions that make up most of the human genome include introns, regulatory sequences, telomeres, and satellite DNA, among others. While they don't directly code for proteins, non-coding DNA plays crucial roles in gene regulation, chromosome structure, and other essential cellular processes. It is not just junk DNA as it was long believed to be. Therefore, a DNA strand is made up of genes and the intergenic region. Within the gene, there are two regions: the exon, the region of the gene containing the coding sequence of nucleotides. The other region inside the gene is called the intron, which is the non-coding sequence within genes. The non-coding intergenic region contains regulatory sequences, which include promoters and enhancers. These control gene expression by influencing where and when a gene is transcribed. In addition, the telomeres in the chromosomes, which are protective caps at the end of chromosomes that prevent DNA damage and chromosome degradation, also make up the non-coding part of the genome. Some of these non-coding repetitive DNA sequences are also found in some other regions of chromosomes, particularly near centromeres, and are called satellite DNA. Now, how does the genetic code turn into a protein that defines our traits? The flow of genetic information from a gene to a protein follows the sequence DNA to RNA to protein, a process often referred to as the central dogma of molecular biology. This involves two main steps: transcription and translation. In the process of transcription, the information encoded in a gene is copied into a messenger RNA or mRNA molecule. This process occurs in the nucleus of the cell. This is the first step where the cell copies a gene's DNA instruction into a similar molecule called RNA. Think of it like copying a recipe from a cookbook. An enzyme called RNA polymerase reads the DNA and builds the RNA strand, which happens in the nucleus. One important thing to remember here is the process called splicing. This splicing process involves the removal of introns and the joining of exons to create mature mRNA, which is ready to travel out of the nucleus to the ribosome. Next, in the process of translation, the mRNA molecule then moves to the ribosome, where the genetic code is read and the corresponding amino acids are assembled into a protein. In this second step, the mRNA recipe is used to build a protein. The ribosome reads the mRNA codons one by one, matching each to an amino acid brought by transfer RNA or tRNA. These amino acids link together to form a protein, like assembling ingredients into a dish. Essentially, the DNA acts as a template, the mRNA carries out the instructions, and the ribosome translates those instructions into a functional protein. So many of you might have heard the word codon. A codon is a set of 3 nucleotides in DNA or RNA that acts like a code word. Each codon tells the cell which amino acid to add to a protein or signals the start or stop to start or stop building it. There are a total of 64 codons.
Aria, there are people who are waiting to join the meeting. Can you accept them? But you can keep going with your presentation.
Go to the meeting. This one. Yeah, just say admit all. Go back to your slides. Yep. Good. Put it full screen. So now I'll investigate gene mapping and gene sequencing, what they mean, how they're different, and why they are important. What is gene mapping? It is the process of determining the location and order of genes on a chromosome. It is like creating a map that shows where specific genes are positioned along the DNA strands. As shown on the picture in the right, gene mapping helps scientists to understand how the genes are inherited and how they interact. How does heredity work, and what role does crossing over play during meiosis, and so on. Gene mapping also helps to identify genes linked to traits or diseases like familial ALS, and then to develop targeted treatments or genetic tests. Another aspect that we should try to understand is the nomenclature used in gene mapping. As you can see in the picture on the left, there are 2 main arms of a chromosome. Note that a chromosome is made up of 2 homologous chromatid pairs. The longer arm is called the Q arm, and the shorter arm is called the P arm. Now, the chromatid is divided into regions. The numbering starts from the centromere and goes on to the telomere. Within a region, there are further divisions called bands and then subbands. Therefore, 3p22.1 would identify the location of a gene as located on the 3rd chromosome on the smaller p arm in region 2, and in band 2 and in subband 1. So now let's look at gene sequencing. Gene sequencing is the process of determining the exact order of nucleotides, A, T, C, and G in a segment of DNA or an entire genome. It's like reading the complete instruction manual of a gene or organism to understand its genetic code. Gene sequencing can help identify mutations or variations linked to diseases, study evolutionary relationships, and enable personalized medicine by analyzing an individual's genetic makeup. In short, gene sequencing decodes the DNA sequence, providing a detailed map of genetic information. Therefore, the main difference between gene mapping and gene sequencing is that gene mapping identifies the locus or location of genes and their relative distance within the genome, whereas gene sequencing spells out the order of the nucleotides, which makes up the genes in the genome. Furthermore, gene mapping results in a less detailed outcome, while gene sequencing results in a fully detailed outcome. So now that we know genomics, codons, and gene mapping and gene sequencing at a high level, let's look at gene editing. Gene editing is a technique that allows scientists to precisely modify an organism's DNA by adding, removing, or altering specific genes. It is like using molecular scissors and a paste tool to edit the genetic code. The most common method, CRISPR-Cas9, uses a guide RNA to target a specific DNA sequence, and an enzyme, Cas9, cuts it, enabling changes to be made. This technology helps to correct genetic disorders, enhance crop resistance or livestock traits, and study gene functions in research. It is a powerful way to rewrite DNA for health, agriculture, and science. Another word you might have heard is CRISPR. The development of CRISPR genome editing has revolutionized scientists' ability to edit the genome. In 2012, research by IGI founder Jennifer Duanda and Emulin Charpentier and their teams developed a method of repurposing a bacterial immune system called CRISPR, an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats, to make breaks in DNA at precise locations using a CRISPR-associated protein or Cas9. Cas9 is like molecular scissors to cut DNA. When DNA is cut, it creates an opportunity to change the DNA sequence and add around the cut side by harnessing the cell's natural repair pathways. So now let's look at the future of genomics and how AI will impact this. The image from the WHO website visually summarizes the impact of genomics on healthcare. It highlights some of the key themes that would predict the future of this field. Genomics will transform from a research field into a medical tool, especially with recent advancements like CRISPR and Cas9. It will pave the way for personalized medicine, tailored treatments, disease prevention, early detection, and even pathogen surveillance and tracking outbreaks. Now imagine the possibility that AI could help as a smart assistant to researchers and scientists in this area. With its advancements, with an advanced computer power, AI can analyze massive amounts of genetic data, find patterns with changes linked to diseases like cancer or rare conditions. AI tools can quickly analyze genomic sequences to spot genetic issues, predict diseases, speed up diagnosis, and personalize treatments. In conclusion, the future of genomics promises revolutionary changes in many areas of life. Advances in sequencing will make genetic testing faster and more affordable, bringing personalized healthcare within everyone's reach. Treatments will increasingly be tailored to individual DNA, while gene editing tools like CRISPR may offer cures for previously untreatable genetic diseases. AI and big data will help uncover hidden patterns in our genomes, leading to new insights. Beyond human health, genomics will improve crops, aid conservation, and drive innovation in medicine and biotechnology. However, with these breakthroughs come important ethical challenges around privacy, fairness, and responsible use. I hope you enjoyed the conversation. I had a great time investigating the topic and creating awareness around it as part of Youth Team Advisors. Thank you. Okay, so I also have a Google form that you can answer to just test your knowledge on the presentation. Can you see it? I put it in the chat. If you also have any questions, you can put them in the chat and I will respond as soon as I can.